Okay. Wow, attendance is up. That must be because the exam is over. And everyone stop panicking. Or you'll continue to panic all week. Okay. So what are we talking about? Gene expression, transcription, something like that. I don't even know what happened to this weekend. It was just like the weather got nice and time disappeared. Laundry didn't get done and the food shopping. My kids have been starved and have very close. Don't ask about I brought home all 75 of your exams to grade. And I won't tell you how far I got. Probably as far as you guys got into studying for the next exam. Right? Okay. So, exam 1 will be graded by Friday, I promise. Oh, you're right. That's exam 2. Monday. Okay. Gene expression topics are due in recitation this week. Be prepared to defend your choices. I'm getting a lot of good questions over email. That makes me think people are engaged, maybe, or interested, or scared. One question I got was about whether or not. So I kind of went on and on about how we shouldn't share our topics with other groups because novelty is worth 10% of the grade. So don't, if another friend says to you, hey, we have no idea what we're doing, what should we do, what are you guys doing? That's probably not a wise idea to share. But I got a question about that, whether that means you can't share within your group. No, this is a group project. You must share within your group to survive. So share within your group, not between groups, in case there's confusion there. Other questions I've gotten, that was really the one that I thought was applicable to everyone. But please feel free to see, people are stopping by all the time now to ask me what I think. And I'm happy to give you feedback. Plus, I'll be giving you feedback in recitation. So there's no paper this week in recitation, but there's a transcription drawing exercise. So we'll have colored pencils and paper, and we're going to draw out transcription in periodic cells and eukaryotic cells so you guys can start to visualize how all these different proteins are coming together. And there will be a quiz. So best to be fully engaged in the exercise. Yes?
If you don't like our topic, we can repick.
Yes, if I don't like your topic, you can repick. Yeah, that's the point of you telling me this week, so that you don't go for 3 weeks studying, working on something, and have it be a disaster, because there's definitely some things that are not worth working on. Yes?
Will you tell us if the topic is overlapping? Like, we'll get the novelty points?
Don't worry about that. Those points are mostly there as it's like carrots and sticks. It's a stick to prevent everyone from cheating and sharing the same 3 topics. Yes, if someone in Friday's recitation has the same topic as someone in Monday's recitation, but just because the topic is one gene doesn't mean the whole project's going to be the same. You should be fine. But if projects start to look similar, I can get rid of those novelty points. And so that's my stick. I hate to have sticks. I prefer carrots, but whatever. Okay, so no office hours today, but there will be the ability to email me to make an appointment. So if you were dying to see me today, email me and we'll work out sometime during this week, or you can come Wednesday or Friday. That's it for business. Any questions about anything? Business-wise? Yes?
Is the recitation schedule that's online still the same?
I believe so. The second one. Remember, we had a revised syllabus, which should have a second. Check to see if the revised syllabus has the new recitation schedule in it or not. If it has the exams on the correct dates, then it's the same. We're just going to be, basically from now until the end, it's this project that's going to take up the bulk of your time. And we will have these small kind of drawing exercises in recitation. And that's because it's a good opportunity to draw things out and really think about them, because there's a lot of different proteins that are coming together. Okay? Okay. So we started talking about gene expression, and we talked about the different types of RNA. I hear a lot of talking. Who wants to tell me what's going on? No? Okay. There was a thing on hearing aids on NPR this morning about how, like, old analog hearing aids, like, increase the volume of all noises equally, so background noise was really distracting for people who needed hearing aids, but now the new ones are really good, and they can actually get rid of the background noise and increase the noise of the person talking to you. And sometimes I feel like I need those. Because I hear background noise whenever people are murmuring, I'm like, what are you talking about? So, anyways, hearing aids. Okay. Many kinds of RNA, not just coding. RNA is made in different quantities by many RNA polymerases, transcribing one gene simultaneously. This week and next week we'll be learning about how that's regulated, because a huge part of gene expression is regulating the level of expression that a gene will be expressed at. That's a lot of express, yeah. Similarities and differences, DNA versus RNA, DNA polymerase versus RNA polymerase, and we did the transcription cycle in bacteria. So today we're on to eukaryotic cells, which is significantly more complicated, but not really. I mean, yeah, it's not just like two proteins involved, but the whole thing is pretty much the same when you take general transcription factors and call them sigma factors. Okay. So let's go on. So this is what initiation looks like in eukaryotic cells. Initiation of transcription of a eukaryotic gene requires assembly of general transcription factors at the promoter, followed by recruitment of RNA polymerase. So you need something to recognize the promoter and recruit RNA polymerase. That's the main big picture to keep in mind. It's all about recruiting RNA polymerase to the promoter of the gene that needs to be transcribed. And there's all these TF2 proteins that are involved in that. Okay? Big picture. So now we'll go little picture. So the first thing that needs to be done is you need to recognize the promoter. And that's very similar to what we saw with bacteria, right? But here you're recognizing the TATA box, which is part of the eukaryotic promoter, by binding to the TATA binding protein. Really easy to remember, right? TATA binding protein binds TATA box, which is a subunit of TF2D, and then you recruit TF2B. So here's your DNA and your start of transcription, and you've got your promoter just before that. And this is your TATA box, which means it's a sequence of DNA that likes to bind to TATA binding protein. Yes?
I was going to ask, why isn't the TATA box like the sigma factor in bacteria?
You are a little sick.
Yes, ma'am.
Sorry. I get that for weeks. Anyways, why isn't what?
The TATA box, the same as the sigma factor.
Why isn't it the same as a sigma factor? TATA box is in DNA. Are you thinking about why isn't the element in bacteria called a TATA box?
No, the question on the is,
Oh.
you know how it said what is analogous to the sigma factor? Why isn't it the TATA box? Aren't they very similar?
Sigma factor binds to the promoter. TATA box is a DNA element.
Okay.
Yeah. So that was about the class this weekend. And about whether or not the TATA box and sigma factor are similar. They're not, because the TATA box is the DNA that's bound by the analogous TATA binding protein, which would be analogous to the sigma factor. Yeah. Next time, walk up here and talk to me. I feel like I'm going to have to give you a throat transplant or something. Okay. So you get this TATA box, which gets bound by TF2D, which has two subunits, one of which is the TATA binding protein. So it binds, and then TF2B is involved, and it binds, and that's the role. So, okay, before we start going through alphabet soup, I want to apologize for the alphabet soup. And I want you to know I hate it too, because really, people were not very inventive in their naming of proteins. They should name them with the function, right? TATA binding protein. Okay, we get it. Binds TATA box. It's easy to remember. But it starts to be transcription factor for Pol2. Give me a letter, which is kind of a pain in the butt. Yeah.
Is the TATA box the same as the
Origin of replication.
No.
What's the?
Okay, so right now you're confusing replication and transcription.
I was just trying to relate both of them. Like, what's
No
the-
relationship. No, transcription is involved in replicating your genome. Not transcription. Replication is involved in replication of your genome. And Tadabox is involved in starting of transcription.
They are analogous. Like, that's where the proteins get recruited to start transcription, where the origin is the protein recruited to start
Okay, so
replication.
analogous in the start of the process, but zero other relationship. So I would even say, don't even think of that because you're going to start relating replication and transcription, and there's no relationship between replication and transcription. And I think I get kind of funny about it, like, you know how dogs, like, their hair sticks up? Like, I'm like the dog with the hair hackle sticking up, because I hate when people get confused between replication, transcription, and translation, and when people associate replication with transcription, so I always want to, like, kill that association. So don't even compare. Don't go there. Bad news. But yes, you're right, it's where things start. Yes. I guess you can make it analogous to a whole lot of processes that have to start. Yeah, absolutely. So the difference is, if I was to put on, I'm going to go with your question then. If I was to put it on an exam, how is the origin different from the Tadabox or the promoter? Then what would you say is a major difference between the origin of replication and the Tadabox in terms of location and function? I know, that may have been a lot. Good morning. So let's talk again. So because I want to say to you that there's a promoter before every single gene, right? That's what determines where a gene starts, is that there's a promoter at the transcriptional start state that's involved with recruiting RNA polymerase. Origin of replication, it's not at every gene by any means, because it has nothing to do with gene expression. It's just a number of times throughout the genome, because that's just the different places that DNA replication will start. I feel like that's one good way to think about it in terms of how different they are. One is per gene, the other is just as many times as needs to be there to copy your genome. Okay, I have confused faces looking at me. Are these confused faces or sleepy faces? Sleepy faces? Oh, I hear you. My son is not sleeping at night again. What the hell? 2.5 years old. That's not okay. He did not read the contract he signed at birth. You do not get born and sleep through the night and then stop sleeping at 2. That's not okay, right? It starts with birth, no sleep, then sleep. Then teen years, where you sleep forever, right? Anyways, I'm sleepy. Okay, so we've got recruitment of Tata binding protein as part of TF2D to the Tadabox, and then that results in recruitment of TF2B. The way to think of these things is find a way to remember their function, not their letters. If you start memorizing letters, you'll make a mistake. B is the second one. I know it's a good way to remember if you have to. It's involved with accurate positioning of RNA polymerase at transcription start site. So remember, that's an important function, right? You want RNA polymerase to be accurately positioned. So TF2D, then TF2B. The binding of the Tata binding subunit of TF2D to the Tadabox actually distorts the DNA. And I thought this was very cool when I learned it. So here's your protein bound to your DNA, and it kinks it. And it's believed to serve as a landmark for assembly of other general transcription factors, because it's the first one, and so it's going to bind and distort it and make it stick out a little bit, and that might help in accurate recruitment of the other transcription factors. Now I say help because it's by no means what regulates it. Okay, isn't that cool? So here's your DNA. So when people start learning about transcription, people always get confused between what is in the DNA and what's in the protein, and it's all about DNA binding or proteins binding to DNA. So remember to keep in your head what is a DNA element, so that means what is a certain sequence in DNA that is bound by a protein, and what is a transcription factor or transcriptional activator or repressor that binds to the DNA at that DNA element. Okay? The DNA element versus protein that binds. And here you've got your DNA element, which is your TATA box, which binds to your TATA binding protein, which is the blue-green there. Okay? Okay, so DNA elements found at eukaryotic RNA pol start sites, right here. There's 4 of them, but usually only 3 out of 4 are present at each promoter. So you don't need to have all 4 to have a promoter, but frequently you do have these 2 for sure. You don't always have that. And these are consensus sequences. So these are the names of the elements. These are the DNA sequences that define the elements. And these are the proteins that bind to the elements. So notice you've got TATA binding protein binding to the TATA box. It's called the TATA box because it's TA. So it is totally fair game for me to ask you the sequence of the TATA box, by the way, because it would be ridiculous if you got it wrong. That might be a giveaway, right? Then there's these other elements with these other sequences. And so you notice the only ones that really bind to the DNA are TF2D, a TATA binding protein, which is part of TF2D, and TF2B. And so those are the elements that they bind to. Okay? So I wrote a question here for discussion. Are these elements and transcription factors involved in regulation of gene expression, and how? What I mean by regulation is is there any specificity? Because remember, when you talk about gene expression, you have to think about gene expression as expressing a gene in response to something, right? Things aren't randomly turned on and off. They're not always on. They're not always off. There's a level of specificity in the regulation. But you'll notice that all promoters tend to have a combination of these elements, right? So are these involved in that specific that specific aspect of gene expression, and if so or if not, how and why? So if you guys want to take a minute to talk to each other about this, then we'll reconvene in a minute. And if you don't know what I'm talking about, it's written right there.
They're all
Yes. Okay, so the real question here is, if these are always here, is this involved in regulation of gene expression? And if so, how? And if not, then what is? Really, right? So, since you guys were chit-chatting here, so who feels really good about their answer? Raise your hand if you feel good. Okay, yep.
I would say something has to activate the TBP TF2D in order to initially bind to the title box, and then the transcription factors are kind of like a cascade after that to start transcription. So I'd say the highest point of regulation would be the TBP and TF2D activation.
Okay, so something needs to be regulating whether or not TF2D or tau binding protein can bind to the tau box, right? Either the protein is activated, something's activating it. Okay, I like that. Who wants to add to that or dispute it? I saw you raising your hand. Yes.
I was gonna say that
Ooh, look at that.
if the sequence, I don't think the sequence is really regulated. I think it's more so the factors and the elements, or the factors, because the DNA is always there. So since it's always there, it's going to be constantly expressed if it's not for these factors. So that's where the expression or where the regulation comes in, is on these factors.
Okay, so the sequence is not regulated, it's the factors. So similar, the factors are somehow being regulated because if they're not and the sequence is always there, how come all your genes in your genome aren't always transcribed? Yeah.
You usually have allosteric inhibitors in these regions that prevent these binding proteins.
Okay, so do you normally have inhibitors in these regions preventing these binding proteins? Okay, so there is a level of negative regulation involved that we will get to. Yep. Not necessarily always right here, sometimes right here. And then you also can think of other ways that we've learned about negative regulation of gene expression already. Yep.
So probably the histones would probably be a part of the regulating because we might be blocking them for when it's time to express the gene and then it will be exposed and then transcription factors will recognize and bind to it.
That is another element that's important. So everything I'm hearing is correct, actually, so far. Histones often might be obscuring these regions, and when gene expression needs to be activated, you also need to activate chromatin remodeling enzymes and all of these other proteins that we've talked about already in terms of chromatin structure and function. We need to activate those in order to move or inactivate certain things, in order to move histones out of the way, to allow transcription factors to bind. Yep, so that's another thing, yes.
Well, because I really disagree with what Howard said.
Okay.
Because he said that sequence is always there, but they are consensus sequence, right? They can be different, even though they are similar, but they're still different. So different sequences will be recognized by different TFTP or other transcription factors that cause gene regulation.
Okay, so these are consensus sequences. Are there different regulators that are going to recognize and regulate gene expression? Not here, but somewhere else, and this is what I'm trying to get out of you guys. Yes?
Does the variability of the consensus sequences dictate how well those transcription factors are able to bind?
That's definitely the case in bacteria we talked about, right? Here, not as much because a lot of the regulation happens at a step before all of this. And so there's a little bit of a hint that I'm giving. Yeah.
What's that in doing in INR?
What is it doing?
Yeah, there's an
It's
in.
binding to TF2D in some cases.
No, there's an in the sequence.
Oh, an N, that means any.
Okay.
N means any, yeah. So the way you read consensus sequences is this could be G or C. This could be C or T. N means it could be any nucleotide. Yep, thank you for, I'm like, in what? Okay. Okay, does anyone want to expand on this? So what we've come up with is there's something that's regulating binding of the transcription factors to the tau box and to the promoter. Something needs to regulate that step because your sequence is always the same. Now there are other sequences called enhancer sequences that we will touch on and we will really talk about soon that are all about regulation of gene expression and about regulating recruitment of these proteins to the promoter. So you can think of it as 3 steps. So what I was trying to do with this discussion, and I think we did pretty well, is talk about where the regulation is coming in. Now I want you to know that these transcription factors that we're talking about are called general transcription factors. All the TF2s are all general transcription factors, meaning they all assemble at each promoter and are involved with recruitment of RNA polymerase for every gene. Pol2. We're talking about Pol2 here. So they're general, meaning there's really not much specificity, and the specificity is in regulation of recruitment of the general transcription factors. And we will talk about that toward the end of this lecture. Okay, any questions about this discussion we've just had? Yes?
I just wonder, are those elements part of the promoter?
Yes, these elements are part of the promoter. Yes, and you might say, well, but that's in the after the transcriptional start site. Actually, elements in DNA that regulate gene expression are all over the place. A lot of the times in introns too. Yeah.
You just said, okay, so that DPE site would not be in an exon.
It would likely, well, it doesn't really matter, does it? Doesn't matter.
I mean, but then it's coding for something also.
So the question is, would the DPE site be in the exon? We'll think about the order in which things happen. So the DPE site, could it be in an exon? Well, why not? Why wouldn't it? So think about the order in which things bind. You have all these things binding, you have transcription. Splicing is a little bit later, right? And that splicing does happen concurrently. But still, this is about recruitment, this is about initiation. This is only initiation that we're talking about. So, but introns are a common place for transcriptional regulators, and I believe there are some in exons too. Okay, yes.
So I just wonder if they are general transcription factors, right? They
Yeah.
bind to any sequence?
No, they'll bind to lead sequence, very specific. TF2B likes this sequence. Tau binding protein likes this sequence. But these sequences are in every promoter.
Yeah, but they will bind to any consensus sequence.
Yes.
And does that mean there won't be any regulation expression involved in this here?
There'll be no what?
No regulation expression here.
There'll be no regulation of gene expression here. Right, because it has to do with recruitment. The regulation is whether or not these proteins are recruited here, not whether or not they. So once they're recruited, you start to recruit RNA polymerase and have gene expression. So the regulation is a step before, which is what we're going to learn about, which is going to determine whether or not they're ever recruited to the promoter. Okay, yes.
I was gonna ask, during replication, does the top box, is that under risk of getting bigger and shorter, and can that harm the cell?
Are you talking, you're saying during replication?
Yeah,
Can you have expansion of a Tata box?
Yeah.
Can that harm the cell? I guess you can have expansion of all sorts of things during replication. Can harm the cell. I mean, there's one per gene, so it might harm one gene. If you have maybe not expansion, but maybe if you have mutation or some effect that's negative, and it's a gene needs to be expressed. But it might be just one gene at a time, right? Not the whole. It depends on what gene it's on. I see your logic. Was that satisfying?
Why are there 30 nucleotides in each element? Why aren't they just like right next to each other?
Think about how big a protein. So the question is, why are there 30 nucleotides between each element? Why aren't they right next to each other? So think about how big a protein is compared to a nucleotide. Right? So these elements are only a few nucleotides long. Let's go back to that picture. See how huge this protein is sitting on top of the Tata box? So if you started to have them right on top of each other, then there'd be no way for it. They'd start to kind of sterically hinder binding to each other. That's a good question. Question here?
Yeah, can you go back to that picture? So if we need these to initiate the transcription, why do we need to recruit more transcription after the start of the
So don't think of it, okay, so if these initiate transcription, why do we need to recruit more transcription factors after the start point? Well, don't think of it as chronological, think of it as setting up the transcription reaction, right? This is all about recruiting RNA polymerase. So all these things, let's go back to this picture. all these things assemble and then RNA polymerase is recruited, then they actually go away. So it's not going to really affect transcription at all, whether or not something binds it. But it is just another binding site to help recruit RNA polymerase.
So the one after the start is going to recruit the transcription factor before transcription starts?
Yes, yes, yes. So don't get confused between where things are on the DNA and the order in which they happen. Okay? I know it's like logically you want to think if it's here, if it's here, something's going to happen before if it's here, but that's not the case when you're talking about recruitment of a complex. It's really just about binding sites. Okay? Getting great questions today. People are waking up. It's warm in here though, huh? Yeah. Okay, any other questions? I feel like I saw another one over here. Are you realizing that you want to take us into left field? Okay, feel free to bring it up in recitation. Okay. Okay, so we've got TF2D and TF2B bound, so we've got recognition of Tatabox, we've got recruitment of a protein that's going to help orient RNA polymerase effectively at the promoter, and then all these other general transcription factors then come in the order that I put. There's T2F that helps with recruitment of RNA polymerase, there's TF2E that helps with recruitment, and TF2H, which is involved with helicase activity, and phosphorylation of the C-terminal domain. I want you to look at this right now. CTD, that's going to be very important, as you've already seen on this weekend's quiz. CTD is super, super, super important in regulation of RNA polymerase and recruitment of processing, all the factors involved in RNA processing. So C-terminal domain just means that C-terminal domain of RNA polymerase is only on pol 2, and it gets phosphorylated, and that affects its function in different ways, and it's a theme that you should kind of keep your eyes on. So now you've got all these factors recruited to the promoter, and RNA polymerase is there. And TF2H is the one with the helicase activity. It's also involved in RNA polymerase 2 C-terminal domain phosphorylation. At least at this point, it gets phosphorylated at many points during transcription, and that regulates the different types of proteins that will interact with the RNA, actually. But at this point, you get phosphorylation, and that changes the shape of RNA polymerase and kicks off all these general transcription factors. And then you've got RNA polymerase, and it's able to leave the promoter and start transcription. So one question I have for you is, I just said TF2H, this one transcription factor, it has helicase activity, it's involved, it has the ability to phosphorylate the C-terminal domain of RNA polymerase. So if I said to you, what domains do you think TF2H might have? What would you tell me? And I want you guys to take a minute to remember domains. I've been kind of harping on this all semester. Think about what domains are. Remember they're a business unit of a protein. There's one protein, one section of a protein that does something. And there's two domains that should be jumping right out at you based on the fact that TF2H has helicase activity and the ability to phosphorylate the C-terminal domain of RNA pol 2. So talk to your friends for a minute. Twice possible. Then that would be comparable. Okay, guys, so tell me, I'm going to use my box list here to ask Kristen Sessa.
Well, we want to run it and
Why a Bravo domain? What do Bravo domains do?
put everything up here, DNA for gene expression.
What do Bravo domains bind to? Yeah, okay, so did I tell you anything about acylated lysine? No, I said helicase activity and phosphorylation of the RNA pol to C-terminal domain. Great idea because, you know what, you were logical in your thinking because gene expression is going to happen. So you went in a logical direction, but you ignored the data I gave you. But, nice try. How about Erica Kutcher? Yes. SH2 domain. What do SH2 domains bind to? Okay, so SH2 domains bind to phosphorylated tyrosine, but they do not phosphorylate. You need to ask yourself what phosphorylates. Let's try for Shawnee Harold. Sorry, up there. You can ask a friend. Ask him or ask her. They'll both be good helpers. Right, okay, great. A kinase domain, right? Because I said it phosphorylates, and kinase is phosphorylate. That was an easy one. Now I see where you went with the SH2, because SH2 binds to phosphotyrosine, but you're thinking the opposite side of the kinase. Kinase is phosphorylate. SH2 binds to something that's been phosphorylated. Yeah. Okay, there's one more. Give me another domain. Yes. ATPase. Do we have any binding of ATP? Okay. Give me logic. She's trying to give me logic for your answer, which is logical. You need ATP to power RNA polymerase. I'm not sure. Go based on the data I gave you. Helicase activity, yes. Helicase domain, right. Now the logic is you might need ATP for that helicase. I forget with this one, but likely. So it's not, you weren't completely far off. Yeah. Because sometimes I give you questions like this on an exam, and you think they're out of left field, but actually I gave you the answers because if you look at helicase activity, you know helicase domain, and if you look at phosphorylation, you know kinase is phosphorylate. Sometimes it's right in front of you. Yes.
Some exam to set ATP hydrolysis domain is like
You might have. If you had said ATP hydrolysis domain and explained your logic, I would be probably, and if you were correct, I would be forced to give you, even though it's not what I was looking for, if it was right, I'd have to give you the points. Yes.
Are those tyrosines that are phosphorylated?
Are those tyrosines that are phosphorylated? It depends on the kind of kinase domain that is in TF2H. I believe serines actually. Not 100%. I think they're serines that are phosphorylated. Okay. Any other questions about this? This is me trying to integrate something you've learned into what we're learning now. How did it work? You like that? Okay. Okay.
It was like trying to think back to like specific
I
domains.
know, you guys think I'm trying to trick you. I'm just trying to see if you can use information you have already. Okay. So here's what eukaryotic transcription looks like in list form. Bless you, and how it's similar. You can think of it as similar to bacterial transcription. So you've got TF2D, which has the ability to bind to Tatabox, and then TAFs, which is the other subunit of TF2D that I put too much emphasis on. And they recognize promoter elements, and it's similar to a domain of the sigma factor, right? Remember sigma factor, there were two different domains of the sigma factor that bind to those two different DNA elements in bacterial promoters. So domains 2 and 4 in the sigma factor. TF2B recognizes another element, this BRE element, and establishes correct orientation. And there's actually a beta loop that goes into the active site, but don't worry about that. And then there's, that's similar also to sigma factor, because there was a domain of the sigma factor that was involved in correct orientation of RNA polymerase at the promoter. That's interesting. Why did they put this one? Okay. TF2A, we have not talked about at all, because it's not even mentioned in Albert's, but it was mentioned in the book I used last year, so don't worry about TF2A. And then RNA, but it's also, whatever. RNA pol 2, obviously similar to RNA pol. So if you notice, there's multiple factors required for the same function as one protein, sigma factor bacteria. So it's bacteria have a much more simple function, but much more simple system than eukaryotic cells. Why do you think there are so many proteins? Oh yeah. Why do you think there are so many proteins involved in eukaryotic transcription in only one simple system in bacteria? Who haven't I called at all yet? And who? So many proteins required in eukaryotic transcription. It's just sigma factor plus RNA pol in bacteria. Okay, so there's a higher level of regulation when we have multiple factors required. I think that's the obvious answer. Yeah. Okay. So notice you've got TF2E and TF2H, and TF2H catalyzes unwinding for helicase activity. Remember, we talked about how that's not required in bacteria, so that's why there's no analogous protein. Yes?
So if you didn't, just hypothetically, TF2B didn't bind with RNA polymerase 2 still binds, it's not correct.
If TF2B did not bind, would RNA polymerase bind but not correctly? Well, I'm not really sure about that, because yes, it's required for establishing the proper orientation of RNA polymerase, but you also need a lot of these other, let's go back here, you need these other factors here to bind, to have RNA polymerase bind.
So does TF2B recruit other factors as well?
TF2B recruits these other factors, yes. So think of it as a cascade, where one step is required for the next step. So TF2B, yeah, you could guess that maybe RNA polymerase would bind but weakly. You could guess that, but these are required to ensure that you have appropriate binding. Any questions? Yes.
Bacteria
Do bacteria not have epigenetic regulation? Like, do they not have methylation? Is that what you're saying? Or?
yes, well, do they have, because the world plasmids in bacterial is that analogous to an epigenetic factor
Okay, so you're saying plasmids in bacteria can alter gene expression. Is that the case? I think of plasmids as just another source of genes that can be shared among bacteria.
So I guess the question is, what is the correlation between epigenetics and eukaryotes versus any kind of semblance of epigenetics?
What's the correlation between epigenetics and eukaryotes versus any kind of something similar, other epigenetics in bacteria? They do have methylation, but I don't know how it works because I'm not well versed in that. Actually, we just had the guy come last week who interviewed for the geneticist microbiologist position. And his whole thing is looking at different E. coli genomes, and he uses this next-gen sequencing method that is able to discriminate between modifications on the different bases of DNA, which is kind of mind-blowing. And so you get your genome plus all the methylation sites. And I was completely unaware of much of anything in bacteria, to tell you the truth. And so there is methylation, maybe not in the same way as in eukaryotic cells, but probably in like the sterics or some sort of recruitment of transcription factors to different genes. Mm-hmm. If he comes here, I highly suggest you talk to him. Okay. Any other questions? Okay, great question. Everyone's awake today. You were sleeping, you woke up. Good. Okay, so there's all these things. Okay, additional protein complexes involved. Two that I want to mention, one that we'll talk about a bit more. Mediator. This is a complex of more than 20 proteins required for transcriptional regulation. So here's your RNA pol and all these general transcription factors and mediators on top. You're going to say, what does that have to do with anything? I'll tell you in a minute. And histone modifying and nucleosome remodeling enzymes, which we spoke about, obviously, right? Here's my mediator slide. Transcriptional activation requires binding of specific transcriptional activators or transcription factors, often far from the transcription start site or promoter, thousands of base pairs away, in the middle of introns at the end of the gene, all over the place. It's kind of mind-blowing when you think about it. And they bind to these enhancer regions. And what they do is they help to recruit the general transcription factors in RNA pol to the promoter. And so that's where the specific regulation comes in. And so as we start the gene expression project, what we're thinking about is gene expression in response to a viral infection. And the viral infection is activating all these signaling pathways that send transcription factors into the nucleus to bind regions far from the promoter in nucleases. And then the DNA actually loops over and then interacts with mediator and is involved in recruitment of all of these general transcription factors and RNA pol to the start site. So it helps distant transcriptional activators communicate with general transcription factors. Mediator, it mediates, right? That's a good way to remember what mediator does, because it's actually mediating the information coming from these specific transcription factors, these activators or repressors or whatever is coming into the nucleus to bind to transcription factor binding sites, mediates that information with the recruitment of general transcription factors and RNA polymerase. All right, I'll let that sink in for a minute as we end class. Any questions about this concept? This is your specificity. You're just confused. Embracing, absorbing.
So, general transcription factors won't assemble out.
They won't assemble without, so the is this guy activated and then you got it flips over and it I just want to say some things might assemble, but really aren't
Putting it off. Fuzzy.